also want to remind people that there's a huge number of posters for this session. We have 17 posters which range through loads and loads of different taxa and loads of different very cool topics, addressing both diversity of the Solanaceae, but also evolution within the Solanaceae. So please do, after we're finished with this session, let's all pop over to that poster session and have a look at those and talk to the poster presenters through whatever channel they've decided to talk to. There'll be a break room at the end of this, and that I will also put that, Susie is going to put that link into the chat at the end of the session. And I'll also put the link to the poster presentations. It's also in your abstract book. All of those links are in the abstract book, so you can find them in there as well. So let's get started, and I'd like to introduce our first and keynote speaker, Dr. Chris Martin, who's from Bucknell University. This is a paper he's doing in collaboration with Andrea McDonnell, who isn't here to present, unfortunately, because I think she's teaching today. So Chris is one of my oldest colleagues in Solanaceae, and I just think that we all ought to give him a gold medal today for presenting this paper in this conference, because Chris not only is doing this from Pennsylvania, where he should be celebrating, but also because he is COVID compromised. So we hope that you will relax after this, Chris, and we're really looking forward to your talk, which is on sexual fluidity amongst the bush tomatoes, the prickly solanums of the Australian monsoon topics. So over to you, Chris.
Thank you, Sandy. And yes, I am chiming in from the now great state of Pennsylvania. Thank you for the introduction, and I'm so pleased to be here. Honestly, isolated in my bedroom is not a fun place to be, but I'm super happy to be hanging out with all you folks today. This is actually clearly a highlight of my day and week. I am going to be presenting some stuff that is related to the spiny solanums of Australia, and a lot of this work is done in collaboration with the aforementioned Angela McDonnell, who's a postdoc at the Chicago Botanic Garden and a former postdoc in my lab. And most of what we're going to be talking about today really has to do with these amazing and beautiful flowers. This is a flower from a solanum from Australia, part of a group we often call the bush tomatoes, part of the larger sort of subgenus in solanum known as Leptostomonum, or the spiny solanums. And today's talk is really going to focus on their flowers and their reproductive biology. Before we get there, I just want to say that so much of this work has been done in partnership and collaboration with lots of folks, including Angela and other postdocs in my lab. My current postdoc, Tanisha Williams, past postdocs, Jason Cantley and Ingrid Jordan-Thaden, all sorts of people that have helped along the way. And importantly, with the collaboration and advisement and guidance of a lot of traditional landowners in Australia. And we have a brand new project that directly ties into the use of certain solanums in Australia by Aboriginal communities in the Western Desert. So we're so glad and grateful to be working with those people. I want to especially point to the fact that everything I'm going to tell you about today was done to some degree in collaboration with students here at my university, Bucknell University in central Pennsylvania. We focus primarily on undergraduate students, but I've also been really lucky to have outstanding master's students as well. So if I say they and we and our, I'm talking about these folks, right? Just so many wonderful people I've had the pleasure to work with over the years. So the group that we've been focused on is really sort of a clade of solanums that occur across the upper third of the Australian continent, a region we might call the Australian monsoon tropics. This is a group we might call the Solanum dioicum plus echinatum group, and you'll see why in a minute. But this is, something like 40 described species that mostly live in two places you see labeled on the map: the Kimberley Plateau in the west, and the top end that is there in the east, that is part of the upper part of the Northern Territory. If you go to these places and you want to find solanums, all you have to do is find the rocks. Most of these are living on ancient sandstone outcrops. So you can find those formations. You can usually find our beautiful plants, and they are indeed beautiful. They do make beautiful flowers like the one I started with, but also are full of gorgeous prickles. And they make really cool fruits, right? Fruits that are anywhere from, pea-sized all the way up to just larger than a golf ball, sometimes with a calyx that is sort of, just barely covering the fruit, and sometimes entirely enclosing the fruits in those prickly sepals, that prickly calyx. So they're beautiful. I've been so lucky to spend time with them in the field, living in and around them. And, lots of prickles in my fingers, which I don't mind because I do love these plants. But the thing that really got me excited about them, that made me fall in love with them in the first place, is the sort of variability we see and the diversity we see within their sexual systems. These 40 or so species live essentially across the same distribution. They are sympatric, and yet they exhibit 3 different, rather distinct sexual systems. And I'm going to go through each of these, including starting with hermaphroditism, which we often think of as the ancestral condition in Solanum. I just got an alert that I am. Can somebody just let me know that they're hearing me? I got somebody that raised hand function.
Yeah, you're being heard.
Okay, thank you. Yeah, I'm just seeing some raised hands here. I'm not sure why. Maybe these will be chat comments. Anyway, back to it. All right. So let's start with hermaphroditism. Among these 40 species, what we see are about a dozen of them that we're not quite sure how they're related to, our study group, or not until now, that exhibit what we might consider hermaphroditism or bisexuality. This is what we see across most of the angiosperms. 90% or so of the angiosperms have a condition where every single one of their flowers has both male parts and female parts. Forgive the use of male, female, but I'm going to do that for clarity. And what we see is they can do exactly what you think they would. Because of the presence of anthers, these flowers produce pollen. That pollen has 3 openings on it, any one of which we can see the emergence of a pollen tube and the delivery of sperm cells. So we have male function present in every flower. Also in every flower, we have the presence of a gynecium, the presence of female parts, an ovary and a style and a stigma. And if we get lucky, right, we have pollination and fertilization, and then the formation of a fruit with fertile seeds. And thus we also have the presence of female sexual function in those flowers. This is what we see across most of angiosperms, and we've got maybe a dozen or so species. All right, so that's sort of standard, what we might consider ancestral within the genus and what we've long thought might be ancestral within our study group. We also have about 14 currently described species in our group that exhibit something called Andromonaceae. And what does that look like? Well, lots of you have seen this before, where in every single inflorescence, we have the presence of a basal hermaphrodite flower at the base of the floral cluster, and then a bunch of males up above it, right? So in this system, these flowers can do also what you think they would do, right? Both the staminate-only flowers and these bisexual flowers at the base of the inflorescence produce a functional pollen grain with 3 openings, and a pollen tube can get out and deliver sperm cells. Male function can be achieved, but only this bisexual flower has female parts. It has an ovary. We can have the formation of fruits with seeds inside of them, and thus we can achieve female function. This is a really cool sexual system. You see it across all sorts of spiny solanums, including in Africa and including even the eggplant that we've probably hearing a lot about this week or today. But if you think that's cool, then you need to sort of hear me out on functional dioecious. So the rest of the species, about 13 described species in our study group, do something called functional dioecious. You may be familiar with dioecious, where we have separate male plants, separate female plants, something we see in cannabis and willow and hollies. But in Solanum, it looks a lot different than those sort of more typical forms. What we see in functional dioecious and Solanum, in particular the ones in Australia, is that we have male plants that have flowers like you see in this upper image, right? And these are the only flowers these male plants make. The only thing that's really visible here are the anthers, and those anthers do what you think they could. They make pollen that has openings and achieves male function. Then the other individuals within these same taxa and these same populations have flowers like you see here. Now this looks just like the hermaphrodite flowers we've been seeing in the previous 2 sexual systems. So yes, it can make a fruit that has seeds inside of it. And because of the presence of anthers, it does produce a pollen grain, but that pollen grain looks quite different from the ones that I've shown you before. And that's because these pollen grains are what we call inaperturate. They have no openings. A pollen tube cannot emerge. We do not have the delivery of sperm cells. So in a functional sense, this flower, even with these anthers and producing a type of pollen grain, is really functionally a female. And that's how we achieve unisexuality in this group of functionally dioecious taxa that look morphologically to be something quite different. This was discovered and described by Greg Anderson and David Simon. They published this in a paper in Evolution in 1989 in this group, right? Greg had previously shown it in some New World taxa in the late 70s, early 80s. They proposed that the way this would have come about, right, that would have been that with hermaphroditism as sort of a backdrop sexual system, we then transition to Andromonaceae, and then Andromonaceae itself would be the transition to this form of functional dioecious. We'd need a couple of steps to get there, right? Some plants would end up with just the male flowers on those individual plants, they would still produce this functional pollen, and now we have male plants, right? Other plants in the same population would end up with just this solitary hermaphrodite flower, right? And then we'd have either the closure or no formation of all of those pores. And voila, we have functional dioecious in Solanum. A lot of this resting on the idea that dioecious within lineages is typically considered an endpoint, right? We get to there, but we don't transition back out. So that's sort of a how it might happen. And that's been sort of the leading hypothesis, those steps since 1989. But if you're like me, you also like to think about why questions. Why would you do this? If you're going to commit, I'm going to personify these plants. If you're going to commit to being unisexual, and some of your plants are going to commit to being female, then what are you doing wasting the energy, still producing anthers and filling them with pollen if that pollen is not going to achieve sexual function, right? And those of you that are thinking about this already know the answer, right? Well, we have a very tight association between pollen feeding bees and Solanum, right? This buzz pollination system relies quite heavily on these bees that are smart, right? And they respond to certain cues. So if you want them to keep coming to visit your flowers, you have to keep your anthers around to be that attraction. And if you want them to keep coming back, you've got to produce some sort of reward. What we see here is essentially a commitment to being unisexual, to being obligately outcrossing all the time. But even in that condition, you cannot afford to get rid of your male organs and some kind of pollen if you want to still maintain pollinator fidelity. It's a beautiful example of a plant-animal interaction that has manifested in these really cool floral morphs. When I learned about this as an early PhD student with Greg Anderson at University of Connecticut, I was absolutely floored and wanted to learn more, right? So I've spent about 20 years working on this group, but 15 of those I've been able to be in the field, 6 different expeditions with wonderful collaborators and people in Australia, getting to know these plants and just living aside of them, right? And also growing them in my greenhouses here and really getting to know these plants. But the issue that I have had over the years is we haven't really been able to resolve a phylogeny that allows us to really sort of unequivocally say how those breeding systems, how those sexual systems had come about. What had they transitioned from? So most recently, I worked with Angela, co-author on this talk, to really try to resolve a phylogeny, a final phylogeny, if you will, to look at the sexual system transitions in particular. And so we sampled lots of stuff, mostly from Australia, our own collections and stuff from herbaria, but also collections of other spiny solanums from around the world, something like 150 or so different samples, lots of stuff from herbaria. Thankfully that those things exist, right? And what we were doing is trying to use a new platform called Angiosperms 353. This is a set of universal probes, 353 gene regions that will work across all of the angiosperms. So part of our goal was to do the sexual system stuff, but also see if this new set of probes would be useful in the Solanaceae. There we are in the herbarium at the New York Botanic Garden. Angela is there along with our former master student Cheyenne Moore and my mom, working in the herbarium and pulling samples for us to use in this study. So I'm not going to go through a lot of the methods. I'm not going to share a ton of trees, but I do want to share a couple of things and then get to the sexual system stuff, right? So we found really decent support for clades within our in-group, all of these beautiful plants being part of that, and some really interesting repeated patterns that other people working in Solanaceae are also finding. Within our lineage, we seem to see a transition from taxa that are sort of wet, tropical, eventually sort of moving into more arid conditions, more disturbed conditions, in particular fire, right? So as Australia has become more and more dry and hot over the last 3 to 5 million years, Solanum, being as good as it is at dealing with disturbance, has sort of moved along with it, right? And we see that trend in our taxa. And so, but mostly we're really concerned about sexual systems. So I'm not going to walk through all of this. I know that was a lot to just open up in one tree, but I want to say we found good support for all sorts of outgroups, stuff that we'll share in the paper eventually. But what about our 40 in-group taxa? What did we see? Well, we see that our andromonaceous species are part of a very well supported clade, as are a set of dioecious taxa representing 2 clades that seem to be quite tightly associated with one another. But one of the really cool outcomes of this work is that our hermaphrodite taxa are part of a clade along with our 2 dioecious clades, right? That they are themselves all sort of part of one well-supported set of relationships. And what does that really mean? So let's look at this tree. This is a tree, follow with me, that shows the potential breeding system transitions and really also shows the endpoints of what the different sexual systems are that we're talking about. So out on the tips here, these colors correspond with this key. So hermaphroditic species are purple, dioecious species are yellow, and andromonaceous species are green. We also have a few taxa that are sort of hermaphrodites, but sometimes act andromonaceous, what we might call weakly andromonaceous. And that's partly what I want to begin to show you is that, our in-groups are these hermaphrodites, these dioecious taxa, these andromonaceous taxa, and then all this stuff that's sort of directly here as outgroups shows a lot of lability. Switch between purple and green and back again, right? Andromonaceae and hermaphroditism, flip-flopping back and forth. It seems like a transition that's pretty easy to make. But the other thing I want to point out to you is that we had been assuming that our sort of ancestral condition working into our in-group was hermaphroditism. And instead, we're finding our hermaphroditic taxa are nested within this group of andromonaceous and dioecious taxa. So what we have here is based on these pie charts that show like the likelihood of any one of these breeding systems being present in this ancestor, is that we seem to have Andromonaceae transitioning to either dioece, as predicted by previous work, or Andromonaceae transitioning first to hermaphroditism and then to dioece. But what's really fascinating is that there's a potential here that if we go from Andromonaceae to dioece, that then we have this unusual, possibly, reversal to the Solanum ancestral condition of hermaphroditism. And what I want to share with you now is whether or not these things are even possible, right? Can you move from Andromonaceae to hermaphroditism? And could you move from dioece to hermaphroditism? Dioece, long thought to be kind of an evolutionary dead end. Once you go to dioece, become unisexual, that's you're not going to transition again out of that. But it turns out there's lots that we can see in the plants that we're growing and spending time with that shows us that it's actually possible. So let's first talk about whether Andromonaceae can switch back to hermaphroditism. Well, we know that's absolutely possible. There's all sorts of work showing that andromonaceous taxa can be, the Andromonaceae itself can be of varying strengths, right? You don't always just have a bisexual flower and a bunch of males. Often, these upper males will have elongated styles and they will essentially transition into hermaphrodite flowers, right? Do that enough and now you're really a bisexual plant. You are hermaphrodite. We see this occurring all the time. The aubergine, the eggplant, is andromonaceous, but you very rarely find male plants up here, right? Most of your flowers are hermaphroditic. So we know when we have longer styles in what should be male plants, we essentially are showing that we can very easily transition from andromonaceae into hermaphroditism. And our tree shows that there's all sorts of lability between these two sexual systems. But what about dioece? Can you go from a dioecious condition where you have unisexuality, males and females, to a new lineage in which you now have hermaphroditic individuals? And the answer is yes, based on what we're seeing with real plants in our greenhouses. What we see is all sorts of leaky dioece, where male plants with male flowers, these flowers will occasionally have fully functional gynecia, an ovary, a style, and a stigma. They tend to be smaller, not quite as robust, but they work. So we are seeing male plants that occasionally produce functionally hermaphrodite flowers. So it actually shouldn't be that surprising to us that you could have a reversal within a dioecious lineage among male plants to a hermaphroditic condition. It turns out we didn't even really need to make the phylogeny to know that that was possible. The answer is right here in these male flowers that occasionally express as bisexual. And they occasionally also form fruits. So this is a male plant in our greenhouse making a fruit, which, based on the fact that it's a male plant, shouldn't be logical, right? So it is absolutely possible for this to occur. But it would matter more if it didn't just happen in our greenhouse, right? Is this just an aberration we're seeing in our greenhouse? And the answer is no, it happens in nature. We see it here in Solanum asymmetrophyllum, male plant making fruits. We see it here with Solanum dioicum out in the Western Kimberley, male plants making fruits. But your question should be, can those plants, can those fruits also actually produce good seed, right? Are they truly doing female function? And we've done all sorts of hand pollinations to show that is possible. The very first plant that we ever used to do this was this species, Solanium2020 from Kakadu National Park in the Northern Territory. And every time this plant made a leaky male, right, that was possible to pollinate, we went ahead and pollinated it, right? Now, a normal fruit's going to look like this, fully formed calyx, 250 to 400 seeds. And our fruits made on male plants had a very poorly developed calyx and only 35 or so seeds, but those seeds did grow, right? At the same time, Greg Anderson at the University of Connecticut also had a species he was working with, Solanium polygamum, a dioecious taxon from the Caribbean. He also had a male plant make some leaky bisexual flowers. He pollinated those. And what we both found was truly fascinating. What we both found is that when you have a male plant that leaks and makes a bisexual flower, and you pollinate that flower, it will form a fruit, it will make some seeds. And if you grow those seeds, every single individual in the F1 generation expresses as just male. Now, this is weird. I've gone and done sex ratios every time I've encountered dioecious taxa in Australia. They're almost always 50-50 male-female. And yet somehow the offspring of a male plant, a leaky male plant, will be 100% male. And guess what? Those males can leak. They will produce hermaphroditic bisexual flowers. And if you pollinate those in your F1s, they will also produce fruits, and the F2s again, 100% male. So here we are showing through these like traditional, like, you know, breeding system, greenhouse kind of reproductive biology experiments, right? That it's totally possible for us, even within that small population, to transition from a just male group of plants to something that is functionally bisexual. Fascinating, right? And we're looking forward to working with folks, including some folks on this that are here today on figuring out why this occurs, but truly awesome stuff, right? So that's a good story. And I just want to share in my last couple minutes that, you know, one of the things we do with our plants and our work is we try to share these stories. We think a lot about science communications in my lab, and that also extends to some of the species we've been able to describe in the last few years, including these 5 species that we recently published in the journal Phytokeys. But I'm only going to very quickly tell you about one because it ties into this sexual system stuff, right? In 2016 and 2018, we collected a mystery plant in the Northern Territory. A thing that we encountered that every time we saw it, different individuals might look like they're bisexual or andromonaceous, or even kind of look like they might be dioecious. And what we found out was that we were not the first to encounter this very population. In 1974, great botanist Pete Latz collected this. And then David Simon, who wrote the monograph of Solanum for Australia, came along in 76. And he said, you know, this has me puzzled also. It's obviously an andromonaceous species, but lacks the racemes of the male flowers. I don't know what this is. It's some aberrant, right? And all those things that were frustrating to them that had them put this one specimen into a cabinet of undetermined taxonomic identity were the things that made us really excited, right? That plasticity, that fluidity in the sexual system is what led us to name this species Solanum plastisexum. And what we did in this very typical taxonomic paper is that we finished with a single sentence at the end, right? We said that this species is really a great example of how in Solanum, in Solanaceae, and in plants, and in organisms generally on the planet, that the notion of a constant sexual binary, black and white, male and female, is actually a fallacy, right? That this is just such a good example, even within a single taxon, of how much fluidity we see in terms of sexual expression. That got us some decent buzz, got us a story in the New York Times. And that meant that, you know, a lot of people ended up going back to the paper. As of yesterday, there have been 9,300 views on the paper. We have this, if you've seen this, our alt metric score is ridiculous for a paper describing a little species from Australia. But my point here is that the sexual system stuff in Solanum is fascinating stuff. It's things that people should be really interested in. And I think this is some proof that they are. But my best example of this is that Erin Johnson, an artist, read the story in the New York Times, and she ended up using Solanum plastisexum as the sort of center of an art exhibit that has now shown in Maine and Los Angeles and in Toronto. And what this says to me is that the sexual system stuff going on in Solanum and in plants in general is just the sort of stuff that can really engage people in our science, right? It makes for a good story. Yes, Solanaceae is great because we eat all sorts of things that are present in this family, but there's all this amazing biology and ecology and diversity that we can also share that people are really interested in. And I'm so happy to kick off this session because you're going to hear a set of talks now that are going to tell you some more of these great stories about this family that we all love. So I'll end it there and say, I really appreciate you hearing my talk today.
Thank you so much, Chris. That was really interesting. I have one question in the question and answer. So we don't have a lot of time, but Antonio Coniglio asked, is it possible that the offspring of these leaky males are actually inbreds and not F1s?
Sure. Yeah. I mean, we don't have a lot to work with, right? So we have a small population of plants in cultivation. And so we are, using what we've got. So yeah, they were siblings to begin with, right? Having come from maybe the same fruits, same population in the wild, and then yeah. So there's all sorts of possible complications there, right?
Okay, there's a question from Chelsea Pretz who wants to know if you have any plans to study the ABCD floral development model, and there's going to be a lot of talks on that in the next couple of days to see how the genes differ between these leaky plants and the strictly dioecious plants.
Personally, no, but we are talking to different sorts of people about potential collaborations to try to figure out the puzzles around unisexuality and selenium.
Great PhD for somebody.
Yeah.
Okay, another question about the andromonaceous flowers. Are they completely, and I think what she means is the andromonaceous, the male flowers, the male function flowers on the andromonaceous species, are they completely carpel free, or do they have aborted or small carpels?
Yeah, right. So fascinating story, right? If we really look inside all of those anther cones of those males, what we see is a very tiny vestigial and typically non-functional gynecium, right? So it's actually not that crazy to imagine that every once in a while, one of those actually develops enough, right? Just enough often. They're not typically very robust, just enough to be functional, right? But they're always there. They essentially never go away.
Okay, and there's a question about, do we know any of the genes that are involved in sex determination? Do solaniums have sex genes, sex chromosomes?
We don't know, right? No on the sex chromosome so far, but maybe regions, right? So this is some of the work that there are folks thinking pretty seriously about right now.
Okay, and then I think the last question we have time for is, if you keep planting the F1 to F10 from the male flowers, do you think your plants would be complete hermaphrodites eventually?
I don't know, I mean, we're out to F2. Yeah, I mean, that's the theory. Theoretically, yeah, eventually we get to a fully hermaphroditic population. That's the way it would have to work. But yeah, maybe I'll keep doing it, right? I could be 80 and still doing it.
Plenty of time. Plenty of time.
F100.
Chris, thank you very much for a really entertaining talk, and thanks everybody for your really interesting questions. And if we haven't got to them all, I'll try to keep them in the chat and see if we can get to them. And then we also have a discussion session after the end. So I'd like now to introduce our next speaker, Matt Gibson from Indiana University in the US, and he's going to talk to us about and now for something completely different, is reconstructing the history and biological consequences of a plant invasion on the Galapagos Islands. Solanaceae grow in the most amazing places. So thank you, Matt. Over to you.
Thank you, Sandy. Okay, can you guys, let me see here. Do you see the presentation or presenter view right now?
No, you're perfect. Absolutely.
Okay, great. Well, thank you everyone for coming to this talk. This is my first time at the Solanaceae meeting. My name is Matt Gibson. I'm a PhD candidate at Indiana University, and today I'll be talking about some of our recent work on the Galapagos, reconstructing the history and consequences of an invasion. So like all evolutionary biologists, I'm interested in understanding the origins of diversity, and the wild tomatoes, Solanum section Lycopersicum, are a really fantastic system for this. I probably don't need to justify this to a lot of you guys, but let me just remind you that there is an abundance of phenotypic diversity across multiple trait axes. The system is genomics enabled and really quite tractable. The group is recently diverged, and as a result of this, there are several potential sources of trait variation. One of these is introgression, which data suggests is actually quite common in this clade. And I think in general, over the past 20 years, the field has begun to realize that hybridization, at least between closely related species is reasonably common, affecting approximately 25% of flowering plant species. However, the role that introgression plays in shaping patterns of adaptive phenotypic evolution is definitely less clear, including in the wild tomatoes, but is arguably the most interesting. So today I'm going to be talking about this charismatic case of phenotypic evolution, which we believe to have been driven by introgression. I'm going to be talking about invasion and fruit color convergence on the Galapagos. So on the Galapagos Islands, there are two endemic tomato species that are described, Solanum galapagense and Solanum chesminiae. However, it's also home to two invasive species, Solanum pimpinellifolium and Solanum lycopersicum, which were thought to have been introduced by humans. So in 2018, I was down there making collections, and we made this interesting observation, and that is that select populations of pimpinellifolium on Santa Cruz Island, the most populated island in the archipelago, are actually polymorphic in their fruit color. About 40% of plants in these populations have orange fruits rather than red fruits, and these orange fruits really closely resemble the endemic species. So immediately the question became, what is the source of this trait variation? Has it evolved de novo post-colonization, or perhaps via introgression from an endemic species? It could be ancestral variation that is just now being sorted onto the islands. However, we have prior data to suggest that that's probably not the case. So today I'm going to be evaluating these two hypotheses here. So this is the setup, and I'm just going to jump straight into the methods. Our first step was to establish some context, right? Where did these polymorphic pimpinellifolium populations invade from, and how many invasions were there? So to begin our analyses, we made novel field collections from the islands, combined them with previously sequenced continental accessions from actually my first dissertation chapter, and then performed some simple as well as model-based inferences of their origin and demographic history. We were able to sample 174 individuals from the islands from 13 populations, which were then combined with 132 georeferenced pimpinellifolium accessions from a previous study, my previous chapter. And then we subject them to ddRAD sequencing with PSD1 and EchoR1 enzymes. I won't get into the details here, but the data turned out pretty good. We went a little overboard on the sequencing with average depths around 61x. So what does this data tell us? Where are invasive pimpinellifolium derived from? Looking simply just at genome-wide divergence, we see this clear pattern suggesting that island pimpinellifolium are more closely related to Ecuadorian pimpinellifolium than Peruvian pimpinellifolium. And then taking a more spatially explicit approach to identify likely source localities based on average pairwise divergence. Basically, we take each accession and then look at how similar it is to, on average, to all our invasive pimpinellifolium samples from the Galapagos. And again, there's this clear indication that Galapagos PIM resemble Ecuadorian PIM. And it specifically points to central Ecuador, where it's the most red in this plot here, as a likely source. Next, we were interested in evaluating the likely number of introductions. So to do so, we performed some dimensionality reduction with the principal components analysis, as well as this model-based analysis using the machine learning software Locator. And I'm not going to go into the details here. However, we have evidence for at least 3 introductions of pimpinellifolium onto the islands. However, only one of these introductions, shown these brown circles here, seem to be responsible for greater than like 98% of our sampled plants. And using Locator, we can pinpoint a likely source region in South Central Ecuador near Guayas and Los Rios provinces. Okay. So our polymorphic populations were derived essentially from a single recent introduction. Although we have evidence for 3 introductions, all of our polymorphic populations and most samples in general were derived from this introduction from Central Ecuador. So these 2 hypotheses remain. So our next step was to evaluate evidence for hybridization and introgression in an attempt to understand the source of this phenotypic variation. And to do this, we employed an ensemble of methods, fast structure, tree mix, new hybrids, as well as our own custom local ancestry hidden Markov model. Modeling migration with tree mix, the most likely network topology implies 3 separate admixture events between Pimpinellifolium and Chesminiae, including between populations which are polymorphic for fruit color. However, when we conduct a similar analysis with the structure algorithm, in fact, no Chesminiae ancestry is detected within these polymorphic populations, populations 114 and 117. And in fact, the strongest signal of admixture we see is evidence for first and second generation hybrid offspring between Pimpinellifolium and Chesminiae at the sympatric site 115, which is on Baltra Road on Santa Cruz. We see clear evidence in the structure plot for admixture, and new hybrids predicts many of these individuals to be first or second generation hybrids. So this analysis definitely does not suggest that orange fruits are the product of introgression, but does seem to suggest, or does definitely suggest, that hybridization is actively occurring, right, within the past 1, maybe 2 generations. So why these different results? We have tree mix telling us that we have all this admixture between Chesminiae and Pimpinellifolium, but structure seems to say something different. So we next consider the possibility that maybe these methods are just picking up on different signals or different signatures. Structure might be capturing these recent hybrids quite well, whereas tree mix might be capturing potentially older introgression events. So to get at this, with the help of Yaniv Brandwein, we wrote some software, a hidden Markov model, to essentially paint the chromosomes of focal populations or individuals from focal populations. We paint their chromosomes with inferred ancestries. Basically, any window in the genome can be of Chesminiae ancestry, Pimpinellifolium ancestry, or of heterozygous ancestry. I'm not going to get into the weeds on this one. However, we did spend a lot of time making sure that it was making valid and accurate predictions, and I'm pretty confident in these results. So applying this to 3 Pimpinellifolium populations on Santa Cruz, we see that average Chesminiae ancestry proportions are indeed higher in these polymorphic populations compared to non-polymorphic populations. And this is what the data look like as inferred by our HMM. We can see for the 2 polymorphic populations, that is, we see that there are varying signals of endemic ancestry across the genomes of individuals from both of these populations. And these introgression blocks reflect older hybridization events. We can attempt to estimate the timing or the age of these blocks with questionable accuracy, and we get estimates in the range of 4 to 12 generations. So still quite recent, but definitely older than the F1s and F2s that I previously described. To evaluate whether any of these introgression blocks may help to explain fruit color variation within populations, we took an admixture mapping approach. We obtained the genomic locations of several key carotenoid loci, a pathway which is pretty well understood, and then performed admixture mapping to test for associations between ancestry and color. Looking first at population 117, we performed this admixture mapping and found that only one locus was predictive of ancestry, and only one locus was predictive of fruit color, and that is the locus Psych B. As you can see here, Psych B is interesting for a couple of reasons. One, it's responsible for converting lycopene into beta carotene. It's a key component of the carotenoid pathway. But more importantly, orange coloration in the endemic species has actually been mapped to this locus in an interspecific mapping population. And so this beta allele, as it's been called, is dominant at this locus and acts to increase beta carotene levels and lead to this orange pigmentation. And I said it's dominant, and that's consistent with what we see here, in that individuals who are heterozygous for their ancestry at the site or at this gene do in fact express the orange phenotype.
2 minutes, Matt.
Okay. Looking at, we performed the same analysis for this other population, 114, but actually found a different signal of association. In this case, color was predicted by ancestry at this gene Psi1. which was quite interesting. Cy1 is the first enzyme in the carotenoid pathway, phytoene synthase. And while endemic mutants have not been identified, other naturally occurring alleles, such as the yellow flesh allele, which was described in Lycopersicum, I believe, these alleles also produce orange pigmentation. So this is also a promising candidate. So to summarize this section, by mapping the landscape of introgression, we've inferred this unique scenario in which phenotypic transitions to orange fruits and two different invasive Pimpinella folium populations were each derived from introgression at a distinct carotenoid locus. And then to summarize my talk as a whole, we have evidence for 3 introductions of Pimpinella folium, but really only 1 invasion from around Los Rios or Guayas provinces in Ecuador. We have clear evidence for hybridization and introgression between Pimpinella folium and Chesminiae. And based on our HMM, orange fruits in select Pimpinella folium populations are indeed likely derived from introgression from Chesminiae. Now this leads us with the open question, are orange fruits favored on the islands? We now have multiple transitions to this fruit color. And so the next steps that I'm looking forward to doing is evaluating whether or not this is a real case of adaptive introgression. So with that, I'd like to acknowledge all my many collaborators, specifically Maria Torres at USFQ and her lab, lots of help on the Galapagos Science Center, Marcelo Loyola, Unique Brandevane for helping with the HMM, and my advisor, Leonie Moyle. Thank you, everyone.
Thank you very much, Matt. That was really great. That was a whistle-stop tour through the Galapagos. We have a question from Jessica Cooperstone who says, have you or anybody else evaluated the carotenoid content of your tomatoes? And I think she means the hybrid ones, the interspecific, the introgressed ones.
No, that has not happened in these focal populations. I do recall an old Charlie Rick study where either he cited some beta carotene composition measurements or did them himself. They have been done in the endemic species, but not in our focal populations.
Okay, great, thanks. And I have a question as well. Are those hybrid populations the same ones that Sarah Darwin found in the year 2000 on the dump on the Baltra Road?
Yeah, so they are on Baltra Road. However, they are not at that site, but they're fairly close. Yeah, that is honestly something that I've been curious about for a while. I'm not sure how we would answer that. I mean, importantly, she didn't find them as polymorphic.
No,
she described these orange plants and hypothesized that they were the result of hybridization.
Oh, that's really
Yep.
interesting.
Yeah.
Well, thank you very much, Matt. That was great. Okay, so our next talk in this series of evolution and diversity is from Pasquale Tripodi from the Korea Research Center for Vegetable and Ornamental Crops. And you're here, Pasquale, aren't you? Somewhere. And he's going to talk to us about how gene-based genomics highlights novel centers of diversity and routes of migration in pepper in various species of capsicum. So it's a bit of a change of genus now. So over to you, Pasquale.
So can you hear me?
Yes.
Yes, good morning. Great. So, and good evening to everyone. So I thank the organizer for giving this opportunity to talk at the SOL meeting.
If you can put it on presenter mode, perfect, that's better.
Okay,
that's
very good. So, and today I'm going to present this work, which is part of an international effort within the frame of the European G2 Resource Project, where we investigated the global genomic diversity of a gene bank resource of pepper. So why we're interested in gene bank? Gene bank has play a key role to address the agricultural change, agricultural challenges of next decades, such as climate change, demographic trends, and also reducing lands of agriculture. However, these potentialities are still unexplored since not all the gene bank diversity have been studied so far. So therefore, there are open questions and main objectives which we want to address with this work. Firstly, we want to check whether the integration of genomics data into the gene bank diversity can help to give more insight into the diversity in terms of phylogenesis, and also suggest some new routes of migration. But also we want to check whether genomics data can help to give more insight into the management of gene bank resource, and also how the combination of phenotypic and genomic data can give novel information in terms of alleles underlying the agricultural traits. So we start from a big collection of over 10,000 accessions retrieved from main gene banks, European and international gene banks. The collection comprises the most common wild and domesticated species, and for the cultivated one, we have almost 7,000 accessions. The collection has been genotyped using genotyping by sequencing. We use the common pipeline developed by Shire with some modification using two system enzymes. And after filtering, different steps of filtering and quality mapping, we developed almost 30,000 high quality SNP, which have been used for downstream analysis. The analysis of diversity have been done at with two different steps. We firstly investigated the whole germ plus collection. Here is possible to see in this composite panel, it's possible to see the admixture analysis on the bottom, and also PCA and phylogenetic tree. The main message here is that the individuals clusterize according to the species. We also found that the relationship between species was confirmed, but interestingly, we found that several species show some mixture. As example, Caenense and Frutescens. Here it's possible to see the blue and green dot. This is probably due to the high capacity of interbreeding of the species, but also due to taxonomical issue occurring in the past. The most wild species were intermingled between the main group, and we better place some species such as Chaquense. Considering the large amount of cultivated pepper, we perform a second round of analysis only on Capsicum annum. And here is the results of the genomic diversity analysis. We found that accessions clusterize according to main geographical provenance. In this matrix on the right, it's possible to see the similarity in the regions, which is almost high considering each regions, and was maximized for species from Asia, South and Southeast, and from species of Central America. Interestingly, we found that accession from Central America and Asia and South and Southeast part of Asia were almost full, so they just belong to a single cluster, K9 and K2. So we rounded for a phylogenetic tree using an outgroup, and we found that accession from Central America, which is the center of origin of pepper, and the south part of Asia, clusterized close together. Also, following a clockwise direction, we found several other groups according to main regional provenance. In the principal component analysis, here we show the first three principal components. It's possible to see the three main gene pool: Central America, Europe, and East Asia. We go back to this picture in the next few slides. And this first set of analysis confirm what already know for pepper, giving additional insight. But the main question was still open. So genomic data alone were capable to provide insight on new ways of diversity expansion for pepper. So we went back to the phenomic data, and surprisingly, we found that the phenomic data stored in GenBank clusterized the accession according also to geographical origin. Here, this is a plot, a B-plot considering the phenomics data, we found that there is a main group of European, Middle Eastern, and Eastern Europe accessions, but also we found that individuals from South Asia and Southeast Asia clusterized closely to Central and South America. Also, we found that for fruit shape and fruit pungency were in the opposite part of the plot, so which means that probably the selection for these two traits have been done in using different criteria. And in fact, looking at the PCA, we found that individuals from Europe were mostly sweet with bell or blocky shape or conical shape, while individuals from Asia were mostly pungent with elongate shape, which is also summarized in this graph on the right. So this means that we can in fact we can suppose that there are two centers of diversity. One, which is already known, is Europe, but also the east and southeast part of Asia can be considered as a center of diversity. If we look back into the history and look into the way of the way of trades, considering the Portuguese and Spanish travel, but also the Japanese travel and the spice trades, this is look like that to Asia materials arrive from different way. And here is possible, in this brief cartoon is possible to see the way of expansion, and also in each country, in each main region, there have been also increasing selection, which led to sweet types in Europe and pungent type with a conical shape in the rest part of Asia. So there are other 2 points I want to address in this presentation. As I said at the beginning, we want to check whether the combination of gene bank genomics data and the historical phenomic data can give novel insight into allele discovery. So we perform a genome-wide association mapping on different traits, and we found that for 2 traits, for 6 traits, there were a very good association. In particular, for fruit 2 traits, the number of pedicle per axis, and for fruit pungency, we found that these traits were in a region under selection, considering the Tajima values. Also, for fruit pungency, we found that only in Capsicum manum, the value of Tajima suggests that these NIPs are under selection. We found that for fruit pungency, there were pyrophosphatase beta highly associated that is on the top part of the glycolysis pathway, which is not directly involved in the capsaicin production, but if we look at the glycolysis pathway, from pyruvate it's possible to produce capsaicin. So probably this gene is more constitutive, but there is in some way an involvement for the pungency of fruit.
2 minutes.
So yes. And also we want to address and to investigate the implication from gene bank genome management. So we used the identity by state method, and we found several duplication, mostly were duplication between including 2 or 3 accessions. But we found that for all providers, the level of application, the percentage of duplicated accession was almost similar. Basically, we found that the percentage of unique accessions for each gene bank range from 80% to 63%. This also depends on the rate of the rate of distribution of germplasm for each that in the history gene banks had. And this is a practical example in which we found, as example, we found one accession that has been duplicated 20 times. And when we look back into the data from gene banks, we found, this is the practical case, we found 3 accessions very similar. that basically they were the same accession in terms of origin, country of origin, and also the name. So the main conclusion, using screening using NGS data, the variability from GERD Plus Bank can give a very interesting insight in terms of early discovery or management. But also in this case, considering the large number of materials, we have also more additional information regarding the diversification of GERD Plus for pepper. So, and as I said at the beginning, this is part of an international effort which has been possible thanks to all the partners from the G2P consortium. Thank you.
Thank you very much, Pasquale. We have time for just one question, and it comes from Carolina Carizo, who asks if you're considering adding any wild species to this analysis beyond just Capsicum chacoense. Are there any wild species in gene banks?
We use we add some wild species like Patermisium, like Tovari, but the number of accessions were very low. So to make to give any insight regarding the phylogenesis, basically. So we have very few individuals from this species. Okay, thank you. Okay, and I think in the interest of time, we're going to move on now to our next speaker. Thank you very much, Pasquale. It's terrible not to be able to applaud everyone. That's the worst part about virtual conferences. Our next speaker is Dr. Andrea Cocucci, who comes from the Instituto Multidisciplinario de Biología Vegetal and in Cordoba, Argentina. And now we're going to take a journey into yet another exciting genus of the Solanaceae, which many people may not have seen. And he'll talk to us about flower color evolution in the florally diverse genus Salpicroa, seen through the eyes of its pollinator. Over to you, Andrea.
Hi, Sandy. Thank you very much for organizing this meeting. Nice to hear you and see so interesting talks.
Can you share your screen, Andrea?
I will try to do it now.
Okay, thank you.
Let's see. Just a moment.
And if you can put your camera on as well, that would be great.
I don't have camera on my computer. Let's see. I'm not finding the right button here.
There we go. Perfect. And if you can put your camera on so people can see you, that would be great.
No, just a moment. That's not the right screen. Are you see it now?
Not yet.
Not?
No, not yet.
Okay.
There may be a slight time lag. We saw the screen before. There we go. Got it.
Yes, now?
Perfect. Perfect. There we are. Great.
Okay.
We're all on the same page now. Thank you, Andrea.
Okay, okay.
Here we go. Over to you, Andrea.
Thank you. The study of flower color diversification and its causes has interested biologists since the beginning of the evolutionary thinking. Initially, flower color was assessed with human subjective color impressions, such as those in Vincent's sunflowers in the vase. Subsequently, chemical knowledge and eventually physical characteristics of petals were utilized to describe and quantify flower color. For example, different flower carotenoids, anthocyans, and combinations of them provide a rich palette of color variants. Also, anatomical and microscopic features of petal tissues provide additional possibilities to build color mixtures by light addition and subtraction. The result of striking light being filtered and mixed can be accurately measured as a reflection spectrum. This has revealed, among others, that flowers may reflect light that cannot be perceived by the human eye, and possibly represent signals that can be perceived by other animals. However, color is not a property of the colorful objects themselves, and is rather an imaginary product built by the receiver's mind when stimulated by the object's reflecting light. During the past decades, background knowledge has been achieved on behavioral physiology, and equipment has been made widely available that allow the study of color as it is perceived by many groups of animals, including most common pollinators, which are the intended signal receivers of flower color. The study of color evolution with receiver subjective approaches has advanced significantly in the research of bird plumage coloration. Much of these approaches are yet to be applied in the study of flower color evolution. For example, very little has been published on flower color as pollinators perceive them, let alone on phylogenetically informed reconstructions of how flower color evolved in ways that account for pollinator perception biases. With this in mind, we aimed to study color evolution in the florally diverse genus Alpicroa, for which we knew that contrasting color variation and contrasting pollinator themes had evolved. Here, pollination by either moths in green, either insects in white, and hummingbirds in orange is reconstructed. Several questions can be addressed with this approach. For example, is chromatic conspicuousness more relevant in plants that are reliant for pollination on hummingbirds, which fly during daytime and do not have a sense of smell, than in plants reliant on moths, which are night flying and have a well-developed sense of smell? Is color change within the clade explained by evolutionary transitions among visually distinct pollinators? The basics we need to know to study flower color in pollinator subjective ways are the spectral quality of the incident light, which is daylight or twilight according to the different pollinators. The spectral quality of reflected light, which can be measured with a reflection spectrophotometer, and the sensitivity of the pollinator eye. Many insects or insect pollinators, such as bees, wasps, and moths, have a trichromatic vision system with 3 light-sensitive opsins in the UV, blue, and green wavelength bands. Our own vision is also trichromatic, but sensitive to blue, green, and red. Knowing the incident light spectrum, the reflection spectrum of the flower, and the sensitivity spectra of the opsins, we can estimate the amount of photons from the incident light that is reflected by the petals and that stimulates each of the 3 photoreceptors of the pollinator eye. These 3 quantities are called quantum catches and can be obtained as the intersection among the areas under the curve of incident light, reflected light, and photoreceptor sensitivity. Thus, each color variant can be represented by 3 numbers, one each for UV, blue, and green photoreceptors. These 3 numbers, if thought as coordinates, can also be utilized to imagine colors as loci in a 3-dimensional perceptive space. A planar depiction of such a 3D perception space is the often utilized Maxwell triangle, where maximum stimulation of each photoreceptor is at the corners of the triangle, and where even and simultaneous stimulation of the 3 photoreceptors, which gives the pollinator subjective white, is at the center. When thinking on color evolution, one can imagine color transitions to occur as displacements within such perceptive spaces. Some pollinators, such as hummingbirds, may have further refinements of color perception, including 4 instead of 3 photoreceptors. This is because hummingbirds are equipped with UV, blue, green, and red absorbing opsins. For the tetrachromatic vision of hummingbirds, color can be represented in a four-dimensional perception space. Here, one such space is depicted as a pyramid, the 4D version of the Maxwell triangle. If one thinks on the evolution of pollinators' subjective color as displacement within 3D and 4D perspective spaces, it is possible to quantify perceived color change. When studying the evolution of flower color as perceived by pollinators, we face a challenge not presented when studying the evolution of plumage color in birds. This is because for birds, the intended receivers of color signals are the birds themselves, and color vision does not change significantly among birds within the same clade. Consequently, evolutionary displacement in plumage color occurs among birds within a single perception space. Contrastingly, flowers are free to evolve within and across perception spaces of more than one signal receiver, which can markedly differ in color vision abilities. To capture this more complex scenario, we propose to integrate the perception spaces of the main pollinators of Salpicroa, moths, and hummingbirds in a single composite perception space. To this end, we reduce the dimensionality of each space to a single axis, for example, with a principal component analysis. In this way, a two-dimensional perception space can be imagined where each axis captures color displacement as perceived by hummingbirds or moths. We obtained more than 400 reflectance spectra from leaf, corolla, and tube and corolla limb of 13 Salpicroa species and studied their location in one such integrated perception space. In this phylophenosphace, species mean color loci of the corolla tube are represented in the integrated color perception space of moths and hummingbirds. It is evident that evolutionary color displacement took place for this particular flower part within perception domains that are almost completely separated between hummingbirds and moth-pollinated species. However, the corolla limb color loci of the same Salpicroa species appeared more intermingled with no clear separation between pollinator-specific domains. One very deviant locus far up right in the plot corresponds to the single species pollinated by insects other than moths. This suggests that corolla tube and not corolla limb color evolved by plants exploiting private sensory signaling channels of the pollinators. The answer to answer whether changing corolla tube color as perceived by pollinators is explained by transitions among visually distinct pollinators, we studied the association between transitions in pollinator mode and along each of the respective perception axes. The curve at top right shows that even when accounting for evolutionary stochasticity and correcting for possible ancestral trends, the correlation between color as perceived by hummingbirds and pollinator mode differs significantly from zero. This does not hold true for corolla limb color, for neither the vision axis of hummingbirds nor of moths. Consequently, corolla tube and not corolla limb color seems to have evolved in response to transitions among pollinators with disparate color vision. So in conclusion, the evolution of flower color in Salpicroa is driven by the perception biases of pollinators and mainly by the perception of hummingbirds. And there is a clear association between flower color as a trait and the transitions in pollinator mode. to end, I would like to mention that what I have said is part of the doctoral thesis of Ana Clara Ibanez, who defended her thesis some months ago during quarantine. Also, Marcelo More and Alice Sersic have contributed with ideas and help in the field work. And Gloria Barbosa, Segundo Leiva, and Paul Gonzalez are experts in the taxonomy and systematics of Salpicroa, and they have also helped very much. And Gerardo Salazar from the Universidad Autónoma de Mexico has helped us a lot in disentangling the phylogenetic relationships among the Salpicroa species. And that was it, and thank you very much.
Thank you very much, Andrea. Those are amazing flowers. Does I don't see any questions in the chat or the questions, but please do come to the kind of break chat to ask Andrea any. Oh, one new message. Does that mean there's a question? Yes. Javiera has asked, do you know the compounds that give the different color and therefore the pollinator preferences in Salpicroa?
Well, we have not studied the chemistry, but it is very probable that the greenish and yellowish colors that dominate most of these flowers are due to carotenoids. Mm-hmm. However, there is one species, the Salpicroa proboscidea, which is pink or salmon colored. And we don't know there if these are also carotenoids or maybe anthocyans. Mm-hmm. Anyhow, the Salpicroas are able to synthesize anthocyans because some of the species have a black or reddish fruits that are colored by anthocyanins.
There's another interesting topic for future research, isn't it?
Yes.
And Carolina asks, is there any relation to tube length? Is the color, I think she means, is the color related to tube length?
Yes.
Or maybe related to tube length.
Tube length is significantly associated with pollination mode. And also, there's a strong association between tube length and some axis of perceived color.
Oh, that's interesting.
Mm-hmm.
That's cool.
So the moth pollinated species are more brownish or greenish mm-hmm. to our perception, and hummingbird pollinated species are yellowish or pink, and they also reflect much more UV light than the moth pollinated species. That part of the color we are not.
We don't have that bit. Well, thank you very much, Andrea. That's a great dip into a genus that many people may not have seen in the wild. Wonderful flowers. Yes. So we'll move on now to our last talk in the session. And this will be given by Remco Stan from the Technical University of Munich in Germany. And we're back to Solanum again now. And he's going to tell us about using wild tomato species to study diversity and evolution of biotic stress responses.
Yes, thank you.
Andrea, if you could stop sharing your screen and then Remco can start sharing his screen, will be perfect.
Yes, just a moment.
So I'm going to ask the obvious question. Can you all hear me?
Yes.
Perfect.
You're fine,
See,
Remco.
My students will be laughing in their office on the other side of this wall now.
Yeah, they will. Andrea, can you stop sharing your screen
yes,
so that Remco can share his?
Yeah, there.
Thank you. That's great. Remco,
okay,
over to you.
Then. There we go. Slideshow. So yes, great. Is this here? There we go. That needs to go and that needs to go. I think this is not the presenter mode that I wanted. Ah, now I'm also the amateur that I was just making fun of. Why is this not working? Give me one more second. So I'm going to just start talking. So because thank you for introducing me. So I'm going to do something different today. So we've seen a lot of genomic data, and I'm actually not going to share anything on genomics, even though we do a lot of genomics in my lab. There we go. Now I still don't have a full Yeah, there we go. So I will talk about how we can use wild tomato species to study diversity and evolution of biotic stress responses. And people who come from a like molecular plant pathogen interactions field, they will know these kind of things. So this will disappear, don't worry. Plant pathogen defense is basically has been historically divided in two layers, like PAMs, or pathogen-associated molecular patterns, which are these very conserved molecules like chitin or flagellin, like cell wall components, and effectors, which are more proteinaceous and more complex, and both have specific receptors, both lead into signaling. And we now know, after 20, 30 years of working with these concepts, that everything is much more complex. There are interactions, there's crosstalk, physical barriers, preformed metabolites, other kinds of crosstalks, and induced secondary metabolites all affect this. And of course, looking in a lab system, it's kind of easy to disentangle this. But what really happens if you work in a natural system? What do you see in nature? So I'm just going to focus on this immune response after one specific elicitation event. And I'll do this in one wild tomato species called Solanum chilense, and there are very good reasons to work with this species. First of all, because you get to travel to Chile and Peru to actually visit the plants. But most of all, it's because it's actually a really exciting species with a very nice demographic history. So we have a central region, sorry about that, a central region as a center of origin here. And then we had two separate migration events, one to a southern highland area and one to the coast on the south. And there is Atacama in between, so we really have two separate legs that we will be able to use. And my PhD student Parvinder will show this in a different talk, that this really gives you some kind of experimental possibilities as well in your lab. And we have a northern region where we think the species migrated back and is still kind of hybridizing also with its sister species, Solanum peruvianum. And we think that it's a great system because the habitats are just so different. These are just example pictures that we took when we were visiting, trying to find these plants in the southern highlands, for example. Here we see that they really grow in these gorges and they're really sheltered from the environment, which is the bare Atacama Desert, or in these more really nice rocky quebradas. Whereas if you go to the coast, you see something completely different. Everything is much more stretched out. But for some reason, look at this here. There's this tiny plant, this tiny Solanum chilense, just growing there. And every morning, or almost every morning, the fog rolls in, and actually there is some humidity brought there. So these plants, they can thrive. And we think that also pathogens actually like this, so that they're also there. And then if you go to the central region, it looks completely different. Well, on the coast, it looks still relatively similar to what I've shown you before. But if you go higher up to 3,500 meters, it looks lush, it looks green. People are growing potatoes there, right? So there's Solanum chilense, this plant that we previously saw in the desert, is growing next to a potato field. So this must mean that there are completely different environmental and biotic stresses. And this made us hypothesize that this must be the best species to actually test what does it mean, diversity and resistance and adaptability, how can you measure this? So we're gonna do this. And the best thing is that last year we published a paper where Gustavo Arias did actually most of the work on this part, that we actually confirmed the demographic history. So we can now really say firmly, this central region is the center of origin, and this coastal and southern areas, they split from each other, from the central region, 30,000 to 200,000 years ago. So relatively recent. So we really see kind of evolution in progress, and we have two descendant populations. And I'm not really going to talk about this much in this talk because I just don't have enough time, but we're using this kind of information. So this makes it a really cool system. And one of the ways we've used it already, and this is also part of this new phytologist paper that I just mentioned, is that we can actually study gene evolution, and we can actually prove that in certain subpopulations, certain resistance genes seem to be under different evolutionary pressures than in others. So if you're interested in gene evolution, please read the paper. If you're interested in more demographic adaptations, check out the talk tomorrow by Gustavo. And if you're interested in more phenotypic differences on how plants and pathogens interact, you should also definitely check out the talk by Parvinder 2 days from now. So what I want to talk about the next, 5 minutes or so that I have left, is how we want to really link genotype to resistance responses and really try to understand how Solanum chilense utilizes the diversity and the kind of plasticity it has in the populations to adapt to certain stresses, or what this really means. And this is work done also by Parvinder Deep, but also by two very talented master students who really put a lot of effort in doing different measurements. And the work I'm showing is really just a snapshot, because what we want to know is how does quantitative defense response differ within and between populations? And this is tough. This is really tough, actually. I underestimated this when we started. So when we started, we took some plants, different populations, here they are, and we just put a random pathogen on it, an Alternaria that we had in the lab with Fusarium and Phytophthora. And to our surprise, we see really big differences. Some plants are completely resistant, others are completely infected. And we couldn't really find a pattern. So this is actually what we're trying to do now. So we really want to look deeper in the pattern underlying these differences in resistance. So we're going to focus in on Phytophthora because that's my PhD, was on Phytophthora. I like this, so we thought, let's just try. And this is work done by Parvinder, as I mentioned. And we want to make sure that our system is reproducible, that we can really make sense of the data. And Solanum chilensis is very thickeny. It's very hard to work with. Also because there is a lot of phenotypic variation on the plants. The good thing is we have perennial plants, so we can pick leaves and we can do our experiments with them. So we have the same mother plant, so to say, in the glass house for several years. When they turn old, like after 5 years or so, we make cuttings, we grow them again. And Parvinder has done loads of infections, like up close to 200 per plant in different batches of repetitions to really show that some plants, and this is what you see here, some plants get infected about 75% of the time. So you put spores on it, 75% of the time they infect, sometimes they don't. This other plant here is much more resistant. Less than 50% of the time, we see this infection actually happening. And this other plant here is 100% of the time it gets infected. And this is all within one accession number, within one population. So seeds collected from one geographic location over here in Peru. And this kind of surprised us a little bit, especially when you come from the plant pathogen literature. You know of differences between accessions, but you don't know how much variation there is within a population. So we tested another one. This guy's in the south. We know they're genetically less diverse because it's been bottlenecked, these populations, but still we see very clear and significant differences between more susceptible and more resistant plants. So now we want to know what's underlying this. And again, like I said, I come from a plant pathology, like molecular plant pathology background. So if you read the literature, people say, okay, one proxy for resistance is measuring the formation of reactive oxygen species. The more the plant makes upon the pathogen landing on the plant, the more resistant your plant is. Basically, in many papers, people don't even measure whether a pathogen is able to infect, they just measure ROS, because they can do this in a plate reader. So that's what we started with. We measured this generation of reactive oxygen species in our population, this 4330 population from the south, and we see that there is actually a huge variation, which is kind of what we expected. But our surprise, it doesn't at all correlate with the resistance. You have these plants here that have a very high burst, but they're not the most susceptible or the most resistant one. And in fact, there is hardly any difference between our two susceptible and resistant plants. We move to another population and we get the exact same thing. So we have our most resistant plant. This is the same population I showed you before. And our most susceptible plant, there's not much difference, but we have some plants here. This one doesn't make any reactive oxygen species when we put stuff on it. Yet it is not completely susceptible. It doesn't die 100% of the time. And this one here makes loads of it, yet it is not suddenly 100% resistant. So this is kind of the first thing that we realized, that these models that you find in textbooks about how resistance works doesn't apply to any system. It definitely does not apply to Solanum chilensis. So our next question is now, and this is work in progress and it's laborious, it's just trying to figure out what else it could be. So we're starting to measure different defense responses. For example, the production of ethylene. It's a hormone and it's also involved in resistances, especially also against herbivores, but also against biotrophic pathogens. And again, huge variation. Some plants show definitely good amounts of ethylene produced, others a lot less, but we cannot really link this to our induction of these defense responses. And in this other population, this one from the central region, we see the same again. Interestingly, we see some trends. And if we try to compare things on a population level, we kind of could, if we want, could make sense of this. So in general, this one population is a bit more resistant. So on a population level, we also measure higher ROS and we measure higher ethylene. So that kind of could work. But then again, we still cannot explain this kind of variation that we see here, where half of these plants have a very high ethylene production and this other half have a very low ethylene production. Yet that doesn't really correlate if you remember the graph earlier with which plant is more and which plant is less resistant. So we could kind of give it a spin, but it doesn't really work yet. And I think this is the take-home message that I want to convey to you. So we're really looking at an extremely clear multigenic trait where maybe in one case your ethylene response is kind of helping to make your plant slightly more resistant, and in the other maybe it's the ROS burst or something else. And there's a lot of stuff known from literature, like this ROS leads to salicylic acid accumulation. Then we have nitric oxide that leads to the accumulation of different hormones. We have other metabolites that are involved, calcium bursts. So there's a lot of chemical processes and biochemical processes that we know are involved in maintaining this kind of resistance, this basal level of resistance against pathogens. And what we really want to do is try to put them now in this global context. So take home message, clearly multigenic. We have a reproducible system where we have perennial plants and where we're able to measure all these different defense responses repeatedly. And we're starting to learn things that these tomatoes are not typical textbooks, but we need to screen a lot more, and we need to really look also wider in different populations. So we're screening different phytohormones, another graph here, salicylic acid, where we also cannot find any correlations, to really catalog the overall diversity that there is. And then ultimately, sorry, I forgot to say, so one of my PhD students, Lina Munoz, is also trying to set up a system so that we can measure new secondary metabolites that might be involved in these kind of resistances, to then ultimately link it to the genomic data that we already have, that we already used to analyze also the demography of the species. Because only when you have a meaningful phenotype, I think we can study also what is happening. And just by doing a genomic screen, we'll find that there is a lot of diversity in these plants, but it's not going to tell us what that really means. So with that, I'm at the end of my talk, and there's a lot of people that are involved in this. We did a lot of sequencing, and on the side, we have also projects where we really try to link the presence of pathogens, and we try to go to these populations and really try to understand where they grow, how they grow, what they do. So all these people have in some way contributed to some of these results. And with this, thank you very much for your attention.
Thank you very much, Remco. That was a great talk. And Esther asks, are you planning to study resistance genetically?
Yes, definitely. So this is the next stage, but the problem is that this genome is extremely diverse. So we have just too many SNPs to actually make meaningful associations at the moment in this demographic context. We could, of course, make a crossing and then sequence the offspring and do a GWAS to find one trait. But what I would like to try to understand is this trait in this network context, where the upregulation of one hormone might lead to the other, and really this whole complexity. And to do this, I guess I just need to be patient, because there is just not enough money to sequence. We had now 150 plants, full genome sequencing for these. I don't have the money for that. So this will come.
You need a partner.
I need a partner with a lot of money, yes.
Someone asked about Alternaria and asked if that was Alternaria alternata or Alternaria solani.
We find Alternaria alternata most of the time in the field, but actually this trial that we did in the lab was with Alternaria solani, because that's a known pathogen on tomato and potato. Yeah.
Okay, another question from Giovanni is, does resistance to different pathogens show different levels of variation, and does it go up in the same way for all pathogens? So
that is a really good question. And the problem is that these measurements are really laborious to do. So this is all done with one molecule that emulates infection with Phytophthora infestans. We've done a few trials where we put like flagellin or chitin on it, so they emulate either a fungus or a bacterium. And we actually see that these variation is not linked. So a plant that shows a very high ROS burst when we put a laminarin on it to emulate oomycete does not necessarily also show a very high ROS burst when you put chitin on it. So this is not linked, and this seems to be really a kind of standing variation that makes the plant adaptable to different kinds of pathogens. But to really get to the bottom of this, we need a lot more data.
And Magda asks, have you evaluated the spectra of pathogens in every environment? In the lab, you have checked a couple of fungi and bacteria exclusively. And are there, I'm not quite sure what the question is, but the diverse mechanisms, are they related to the diverse pathogen spectra? So it's kind of a question of the spectra of pathogens, but also different environments.
So we've been to most of these Solanum chilensis populations. And initially, we of course wanted to identify Phytophthora infestis because that's my pet pathogen, but we didn't isolate much. And then we started to see what else is there. So we know that there are some Alternaria species, Ulocladium species, Staphylium species, Cladosporium species, but that's about it. Because when you go in this field and you harvest these leaves, first of all, you need a permit to isolate your pathogens as well. And we cannot sample more than 5 leaves per plant from the Peruvian government. And then how are you gonna really isolate all these individual microorganisms and determine whether they're a pathogen? This is really difficult. So we're trying to do some microbiome studies as well, but I didn't have the permission or the time to do this on Solanum chilensis. We've got it from some other wild tomatoes. And this basically tells you that all the known, OTUs that you always find on any different plant, they're all present. So there's loads of stuff there. Just trying to really figure out what is the potential pathogen that the plant needs to adapt to is the next step.
Okay, so I'm going to do 2 more questions, and then I'm going to invite people to come into the break room to ask you some more. The first is, have you any idea what's behind the difference in the accumulation of ethylene?
I do not have any idea. That's a very honest and very quick answer.
And the last one is, have you considered doing RenSeek?
I have considered doing RenSeek, but I don't know which probes to design how to design the probes. So we've done RenSeek, so our gene enrichment sequencing on all the Solanum chilensis already. This is in the new phytologist paper. So all the known resistance genes, we've all sequenced them. And this doesn't show any clear pattern that we can link to quantitative defense responses as you would see with oomycetes. So the next stage would be to do some kind of targeted sequencing on a subset of genes, but seeing that all these pathways are already is such a different level involved, it's going to be really tricky to design your probe set to actually capture this. And therefore, I'd rather wait for a very rich partner that sequenced the whole genome. I don't know. This is maybe not the most satisfying answer, but at the moment we're kind of struggling. But if someone has great ideas, would really like to discuss, because I think this is the next step to really start linking genomics with these readouts.
Okay, great. Thank you very much, Remco. And people who are in the chat will notice that there is a link to the virtual poster session, and I put it in twice. And there's also a link to the meeting room. I'll put that in the chat now. And also, I would just like to take this opportunity that people who are interested in diversity and evolution in Solanaceae, 3 very innovative graduate students have established a weekly Solanaceae online seminar series. And you can find out which have hour-long seminars on a huge variety of topics in evolution of Solanaceae across loads and loads of genera. And you can find out about those in the Solanaceae source website, which I'm just going to put the link into the chat now. And there are some YouTube videos of past seminars that people have given. And every week on Friday afternoon at 4 o'clock Central European time, there is one of these seminars with people from all around the world. So please do join us to learn more about diversity and evolution of Solanaceae. And I'd like to thank all of the speakers from today's session for really interesting talks, showing how Solanaceae can also be a really important system for looking at evolution in the field, and also how there is so much diversity out there, which sometimes we fail to capture when we focus on just a very narrow set of things. So there's a lot of diversity out there. There's a lot of diversity to understand. And what's even better is there's lots that we don't understand yet, which makes it really exciting to do. So I will go to the break room now, and then if the speakers can join, and we can all anybody who has questions for the speakers can ask those. And at 5:45 Central European time, we'll have the poster session. And today there are posters from biotechnology, diversity and evolution, and development all together. And there are 17 of those in that poster session. There are 17 diversity posters. And I think it would be really great to see some of the other work that people are doing across this, because choosing people to speak was really difficult. So thanks everybody for coming. And especially thanks to Chris Martin, who, despite being COVID compromised, gave one of his kind of classic amazing talks. So thanks everyone for coming, and see you in the break room.